Okay, so there's this crazy new DNA editing technology that has the potential to treat tons of different kinds of genetic disease with the same basic approach. If you haven't seen my video on it, it's here. But is it actually a good idea? Let's talk about it. So as I describe in the video, the idea is to edit people's DNA to ignore it when the genetic instructions to build a crucial protein have a mutation that says stop building halfway through. And that's great. You stop ending up with these half-built proteins, your cells work a lot better, and you stop being sick. But as a lot of very smart people in my comments pointed out, there is a stop building step at the end of every gene. So if you tweak your cells to ignore that, won't you end up with a bunch of out-of-control protein building? Well, luckily you have evolved a lot of very cool molecular mechanisms that prevent that from happening. Firstly, there are multiple ways that you can word the command, stop building this protein. DNA that makes up your genes is made up of these 4 letters, and every group of 3 subsequent letters serves as a step of the protein building instructions, or a codon. Most codons say add the next piece of this protein. Stop codons say stop, but there are 3 distinct 3-letter combinations that can function as stop codons, which means 3 different ways that your DNA can say stop building this protein. And at the end of a gene, there are multiple different stop codons in a row, and this DNA editing strategy only teaches your cells to ignore one of them. Secondly, when DNA instructions get printed out by the cell and sent over to the protein building machinery, they get a little extra page added at the back. It comes right after the step that says stop, and it's called a poly-A tail. It basically is just a bunch of letter A's in a row. It's been shown that there are molecules within the cell that can recognize and grab onto that final added page, physically blocking the protein building machinery from reading past the stop sign. But what if it does read that whole back page by accident? That 3-letter combo AAA is codon speak for this protein needs another lysine, and in this IKEA analogy, you can think of a lysine as like a chair leg. So if that back page gets read, you end up with a bunch of extra lysines added to the back of your protein, which is like having a chair with 40 extra legs. So when a protein shows up with way too many lysines, the cell can recognize that that back page got read, the stop step was not obeyed, and they can pack up and get rid of that protein. So firstly, there are multiple different stop signs at the end of each gene. Secondly, those stop signs are more difficult to ignore if they occur close to the end of the gene where they're supposed to. And thirdly, if you do end up making a protein with way too many parts, it's done in a specific way that is recognizable by the cell as a junk protein. And if you're still not convinced, we can just look at the data. These scientists did check to make sure that they hadn't messed up these cells' ability to make their other proteins well. And from the experiments they did, it seems like the other proteins were getting made just fine. Now, as I said in my last video, there is still a lot of testing that needs to be done before we jam this into a person. If you are watching this video and you're like, I still don't think this is a good idea, you might be right. But every medicine starts somewhere, and this is a very promising start.